Hi everyone. In this video, we'll be talking more about the metabolism and energy output and input and all that, and enzymes that are within our cells. So let me just insert something here. That way I have my laser pointer. So, energy and metabolism. Basically, the energy that sustains most of what is known and in the earth, it basically comes from the sun. It basically gives heat energy and it goes into the plants where they use that to create more food sources and all that. Um, Energy to kind of give towards like the consumers, and then eventually it goes into energy into the decomposers where it's all decomposed, and then it returns back to that heat energy. And basically there is a field called bioenergetics where they study the flow of energy throughout a living system. So what does it mean to be what does the term itself metabolism means? Metabolism refers to all chemical reactions within a cell. It can be a series of biochemical reactions that convert one or more substrates into a final product. Like, for example, where energy from the sun is captured during photosynthesis to convert carbon dioxide and water into glucose, and then the glucose is the energy stored in there is kind of released during cellular respiration, recreating that carbon dioxide and water. We'll basically discuss these in the next few lecture videos. So there are two types of metabolic pathways, including anabolic and catabolic. The anabolic ones require energy to make a bigger molecule. Catabolic ones basically release energy and break it down into break down larger molecules into smaller ones like this. So it's like opposite reactions. There are different types of energy. So energy is basically the ability to do work, and it can be classified into two types, including kinetic energy and potential energy. So basically any object that has the potential to move, like this set of water here, where it's still, it's more locational energy and potential energy, whereas objects in motion are more kinetic, where they are moving and the energy itself comes from that. Is the objects in motion end up having a kinetic energy where it's like this waterfall where the water is gushing down with so much energy, basically. So now energy within chemical reactions, these are the same types of energy, where it's kinetic, where it's the energy of motion, where if it's like depending on the thermal energy, it keeps moving around, bouncing off. That's where you have your chemical kinetic energy or thermal energy. At the molecular level, this is actually called thermal energy. Now, like we talked about with potential energy, what happens is that this is the energy of position or configuration. So it's more like within the chemical bonds. It's chemical energy that's more in the bonds themselves. And breaking these bonds ends up releasing the energy in the form of electrons. So basically, what happens is you have the highest potential energy in nonpolar equal sharing covalent bonds, where it's like C and H, where it's the electrons are farther away from the nuclei of the atoms, whereas when it goes unequal sharing, what happens is that like polar molecules, what happens is that they have low energy when the uh, electrons are closer to like the more electronegative atoms. So it's like short, strong bonds here, whereas there are long, weaker bonds with the other ones. So it decreases in potential energy as it goes equally sharing to unequal sharing. Now, with chemical potential energy, what happens is that the electrons themselves are the most important source of potential energy, where it depends on the location or the position of where they are in the electron shells. The electrons in the outermost shells end up with the greatest amount of potential energy. So breaking these bonds end up releasing so much energy in the form of electrons, like we talked about earlier. Now, to kind of figure out whether a reaction itself is spontaneous, meaning that there is no added energy, or if it needs energy to fire, there is something called the Gibbs free energy change. This is an equation where it's like delta G, which is the Gibbs free energy change, equals the delta H, which is the change in enthalpy, uh, minus the temperature in degrees Kelvin, and uh, delta S, which is the change in entropy. So the first thing we're going to talk about within the Gibbs free energy change equation is called enthalpy. Enthalpy is basically the total potential energy of a molecule and its effect on the surrounding pressure and volume. So basically the delta H is the difference in the heat content of the reactants and the products measured in joules or calories. So we have the reactants and the products. It's the difference between the two. If it's endothermic, it means that it needed heat. And then if it's exothermic, it released heat. So that's what this aspect of the Gibbs free energy change shows us. Now there is something called entropy. This is basically a measurement for randomness or disorder in a system. It increases when the products of a chemical reaction become less ordered than the reactant itself. So the delta S would be greater than 0. When if it increases, it increases when no work is put into the system, where this is so like with ice, if it's like work to keep it frozen, if not, it will end up with melted puddle of water, basically maximum entropy to the minimum order. So like ice being ordered, whereas in water, it's like more disorder. So now if the entropy in a system decreases, what happens is that the delta S is negatively charged, negatively numbered, less than 0. So basically the work itself required to put it out of disorder. So the energy must be used to produce a highly ordered state. Now, why do we need all this? We need to kind of figure out if we need energy, if we don't need energy. This basically tells you if a reaction in a cell or a system will actually occur non-spontaneously or spontaneously. So if the delta G is less than 0, it ends up a spontaneous reaction where no energy is required. And this is called an exergonic reaction where the energy is released. So if it's negative, it's exergonic and it's spontaneous. Now, if we have a delta G value that is greater than 0, which is positive, and the reaction itself does not require any energy, what happens is this is called an endergonic reaction. And it must be absorbed, energy must be absorbed for it to go forward. So so negative delta G, spontaneous, exergonic, energy released. If it's positive, delta G is positive, it's a non-spontaneous reaction that needs energy and it's endergonic, and we need to absorb energy for it to occur properly. So now exergonic and endergonic basically refers to the changes in free energy. And what happens is basically the changes here is basically where the Gibbs free energy is from the reactants to the product. If it's exergonic, it's like we said, spontaneous and it releases energy. But then with endergonic ones, it it requires more energy, so it ends up having more energy than the reactants. So just keep in mind the differences between the two types of reactions that we can have here. So now we're gonna go to our favorite little energy currency. This is adenosine triphosphate, or ATP. It's our cellular currency for energy, basically. Why, you might be thinking? It basically provides the fuel for most cellular activities. It has a very high potential energy, depending on how these are. And then stored in these chemical bonds that we have. And then it allows the cells to actually function correctly and do its job. The phosphate group itself and energy of the bonds end up being transferred to that target molecule itself. And basically in the structure, it has 4 negative charges where the oxygens are negatively charged. And basically it repels each other from each other. And ATP itself is unstable, and the bond between the outermost phosphates basically spontaneously hydrolyze, resulting in an adenosine diphosphate, or ADP, and an inorganic phosphate group where it has H's with it, like the hydrogens making the oxygen more stable, and ends up with energy being released, which is basically like 7.3 kilocalories per mole. And that is what happens when ATP is hydrolyzed. So now, how do these cells end up using ATP for work? These end up being used when like enzymes end up using them. So like for an example, it's like using enzymes. Enzymes are basically any protein catalyst that speeds up any reactions. So enzymes can speed up both spontaneous and non-spontaneous reactions. They couple together in uh, they couple energy releasing ones and energy requiring processes, basically to form either like a amino acid synthesizing amino acids or moving motor proteins. So basically the ATP comes in and it helps like other components in the cell and be like, yo, we need to get moving, or like getting a potassium pump or something moving. We have those ATP molecules going and binding somewhere and be like, we got to get moving. We need energy to do all of the work we need to do. Now, there is something called energetic coupling where exergonic and endergonic reactions are basically linked together to get a certain biological process moving. So like this example here is where we have ATP hydrolysis and an amino acid synthesis occur in conjunction with each other. So that's basically we're releasing energy from the ATP hydrolysis, and then that energy is taken up by the glutamine synthesis reaction here and ends up coupling together like what we would refer to as energetic coupling. Now, just to kind of just like see what is actually happening with it in this energetic couple is that the endergonic reaction itself for the glutamic acid turning to glutamine is not spontaneous, which means it's an endergonic reaction where the delta G itself is positive. And then the ATP hydrolysis is exergonic situation where the delta G itself is negative and it's spontaneous because of the instability of this ATP molecule. Now, we talked a little bit about enzymes earlier, but we should talk a little bit more about them because these are basically protein catalysts that allow the catalysis of one specific reaction. You can have 1,000 different enzymes, but each one can only carry out one type of reaction. So like the enzymes bring the reactants together in precise orientations that they can actually bind to the active site. So this active site here, they come in certain orientations, they become that complex. This is the transition state. And then what happens is that this reaction occurs and it forms what is called a product in this case and releases that product. Now, specific characteristics of enzymes is that they speed up chemical reactions like trillions of times faster than without it. So like if it does, and the enzyme itself does not get used up in the reaction, it just stays there, just releasing substrate and protein, kind of the substrate and products. So like these, where it's like being built up. Now, when when these reactions occur at the speed of light, basically, it's like an example where the bread eventually disintegrates into small little molecules, but with enzymes, it can actually happen in like minutes, basically. So like this bread can be a molecule of glucose in a matter of minutes. Now, there are certain cases where an enzyme has to go through conformational changes when certain substrates need to be bound. This itself is called an induced fit, where it allows the substrate and the enzyme to properly match with each other. And what we see here is when the ATP glucose binds to that active site, the enzyme itself changes shape, and this induces a fit, reorients the substrates and binds them together more tighter to that active site, allowing the phosphate to be released from the ATP hydrolysis. Like, for example, for this one here, where the glucose gets activated by like hexokinase, it uses the ATP and allows it to become this molecule here. Now, how do these enzymes lower activation energy? One thing about enzymes is that they speed up the reaction by lowering the activation energy that you would see within the uh, system itself. So what happens is that it positions two of them to align perfectly for the reaction like this. It provides an optimal environment where it can like be in a way that it's like if it's acidic or polar, or like within that active site, it kind of gets adjusted, and then it contorts and stress the uh, substrate, so it's like less stable and more likely to react. So when it's less stable, it's more likely to react, so it can change the substrate to actually get it up and running properly, and then temporarily react with the substrate to kind of chemically change it and make it less stable for it to actually help out. Now, with the activation energy itself, what happens is if we have the substrate here and the protein here, sometimes it requires a lot of energy like this. But then if you include an enzyme, what happens is that this activation energy becomes so much lower, and you have more energy that's released than taken in. So basically what happens is that this is the free energy required to reach that area, but then enzymes lower this area here. Now this is a cool video where you guys can check it out if you guys want to know more about how enzymes work. Now, without now enzymes are wonderful, but if they are not regulated correctly, it can lead to so many issues. So how does an enzyme be regulated? The cells end up controlling the enzyme activity to meet their specific needs. So it can change depending on signals from your environment inside the cell and many, many other aspects. So for an example, cells in the pancreas works harder and produce and release these different digestive enzymes depending on the meals, after meals to overnight when you're sleeping. It really depends on how that's regulated. So like the insulin amount, what happens is we have these, if those enzymes are working, they like there might be more enzymes after a meal rather than when you're like sleeping. Now, these enzymes can also be regulated by the changes in the environment, including temperature or pH, because proteins enzymes are a type of protein, so they love their optimal ranges. If they're not in a specific pH or a temperature, they're gonna be like, why are we here? Nope, we're gonna be denatured. Goodbye. We don't want to deal with you anymore. And it can also be the availability of different coenzymes around it or cofactors, depending on like vitamins, minerals, other cofactors, coactivators, so on. And these can actually bind to the active site and be like, okay, we need to get moving, and end up catalyzing that reaction itself. So now it can also be regulated through inhibitors and activator molecules. This can be through like competitive inhibition, where molecules in the similar size and shape to the substrate ends up competing with the substrate to get to the active site itself. So this is directly on the active site, but when we look at like allosteric regulation, this is basically binding to another corner and just like um distorts the active site, basically, like tells it, no, we're not taking this, we're not doing this. A regulatory molecule comes in and it's like, no, no, we're not doing this, we're gonna switch shapes, sizes, and all that, and deactivate or activate that mechanism depending on the enzyme and the regulatory molecules themselves. So the next thing about them is that these enzymes can work together in a metabolic pathway, where these are basically series of reactions like we talked about with like different anabolic and catabolic reactions, where each part can be catalyzed by enzyme, different enzyme, same enzyme, depending on what the reaction is. The product itself is the substrate for that next enzyme, and then it kind of builds their way to the end product, like a series of steps. And this is basically a way to kind of build or break down a complex biological molecule in a stepwise fashion by using the energy from ATP. It can also be regulated by feedback inhibition. So it could be like a these where the substrate comes in, it activates, it gets the allosteric binding, changes the conformation, intermediate A, next enzyme, all that. And then what happens, the product is that when there's a high amount of product, what happens is that the product itself can give a feedback saying, okay, we don't need any more. The product itself can bind to the enzyme as an allosteric competitor, basically, where it changes the conformation and doesn't allow the feedback, the pathway itself to continue. And with that, in the next lecture, we'll talk about more pathways such as like the cellular respiration and photosynthesis activity. And photosynthesis by plants.